Okay, so what are the tools that I would like to introduce today? So they are Selenzyme, DetSpace, RetroPath, Galaxies in BioCAD. So maybe you might say, oh, there are many things here, but at the end, they are tools that are quite interrelated and they are interconnected also. And that's why my idea is that it's better to show them just how they work between them. And they are all about metabolic engineering and the design, build, test, and learn cycle. That's why the way I'm going to present the tools is first, I'm going to give you some introductory ideas of the context where we are using these tools, which is the design, build, test, and learn framework for metabolic pathway design. And then we will go directly into two of these tools that are RetroPath and Selenzyme for metabolic pathway and enzyme selection. And then next, I will show you how we are integrating these tools through a Galaxy platform that is called Galaxies in BioCAD. And finally, we'll also present DetSpace, which is a more recent tool that we have now added into the toolbox and is useful mainly for biosensor design. So first, as I mentioned, we start with some ideas of where we are and why these tools are important. So you need to think that we are in the context of the bioeconomy. There are different initiatives about replacing most of the materials or chemicals that currently are produced from fossil resources through other tools and products that are bio-based, in the sense that they are obtained from renewable resources using, for instance, microbial fermentation. So we are seeing a lot of interest from different economies on that. There is ongoing infrastructures like I2SysBio for in European industrial biotechnology and synthetic biology. And more recently, the European Commission has been announcing different initiatives and is expected to publish now in October the European Biotech Act 2, which is going to be quite relevant for what I'm going to present today. And then another aspect that also is very important in this context is the emergence now of biofoundries, which are facilities. Usually they are highly automated and they allow for reproducibility, high throughput, automation for different technologies like molecular and synthetic biology, metabolic engineering, systems biology. We have an ongoing initiative here in Valencia, which is the Valencia Biofoundry, that has been recently funded. And basically, what you will find in all of these facilities is this pipeline or workflow that you see here. That was published a few years ago, but still is quite valid when we talk about the DBTL cycle. That is the one that is central for biomanufacturing. So basically, you go through different stages in the design, start by this product or material that you want to produce. For instance, some carboxylic acid, and look for metabolic pathways that are going to be introduced into the or engineer into the chassis organisms, the enzymes that are needed, the DNA parts, the experimental design, and finally the assembly of the strains, which is all done mostly in silico, because it's then at the next step where there is the wet lab part of actually building or implementing those different strains that are then quantified at the test stage that generates omics data, that then is fed back into the learn stage that will use some type of predictive modeling for generating new design rules. So that's the basic idea of the biomanufacturing cycle, very similar to other manufacturing industries. And this is what we wanted to highlight here. And our group has been developing several tools around this design and learn, which have been published in different journals, and also they are open source tools that can be installed separately because each one has a different function. Like for instance, for enzyme selection, for pathway design, promoter selection, but they can be also combined because they talk between each other. And why we need all of these tools? Because basically what we have is the situation where in nature you find pathways like this one here for a secondary metabolite, like a flavonoid that you usually will find in plants, that has important properties that go from therapeutics to, for instance, for material, building blocks, or other type of applications. And in fact, what you can do here from that point is to add other enzymes that maybe are not found in nature in that way, in these pathways, but we create novel pathways with glucosylation, methylation, prenylation, hydroxylation, and by that means what we obtain are novel compounds with enhanced properties. And this is what we want to study. It's a combinatorial problem from the biochemical point of view, and that's why one of our main strategies here has been to look at the chemical transformations as some sort of a mapping between graphs or subgraphs. If you look at the molecule, what you can do is to decompose it into basic fragments, and those fragments define some fingerprint or signature of the molecule, and we can do the same for the reactions. Here, what we publish is a database that is called RetroRules that contains, I think, around 500,000 of representation of reactions using this encoding that you see here, and it's called SMARTS usually, or SMIRCs as well. And we call these reaction rules that can be used as a way of generating new or novel putative compounds that can be processed by those enzymes represented by these means. So this is our basic technology. And then let's look at how this can be used for metabolic pathway and enzyme selection through RetroPath and Selenzyme. So as I mentioned, there are plenty of tools. Here, this is a survey that we carried out a few years ago of when going into the different steps at the design stage, like for instance, enumerating pathways, evaluating them for toxicity, yield, number of reactions, performance, ranking, or building them by looking at the DNA parts. There is a large collection of tools that are available. So we need a way of having them integrated. And let's see, first of all, one of these tools, which is RetroPath 2.0. What it does is to look at metabolic pathways that can connect some target compound or source compound of interest for you, because it's one of these chemicals that is useful, and we want to be able to have them produced in a microbial fermentation system or process. And for that, RetroPath, what is going to apply is these reaction rules that I mentioned before, looking for pathways in a retrograde way that can connect this compound with the what we call the syn compounds, that are the compounds or metabolites that are available in the metabolic network or your host organisms. For instance, it could be a bacteria like E. coli or Saccharomyces, a yeast. So this is what we call a retrosynthesis. Here we have a video of how this was done. Let's see if I can make it okay to work. Okay, well, wait a minute. Well, anyway, if I cannot make it, Anyway, this is a demo showing sorry, about this demo of how this RetroPath tool was originally implemented. We use NIME, maybe you know, is a workflow system, okay? That basically you define, like here, nodes that perform different tasks. In this video, you see first that we define our target, that was a terephthalic acid, then the collection of reaction rules that are going to be fired, then the syn compounds, the compounds that are in the host organism, and then after that, the rules are fired. And with that, what we obtain is a network, what we call the scope, the metabolic scope you see here. And the metabolic scope is basically what connects the target compound with the metabolites that are in the chassis organism. So for every step here, we can look in more detail at the compounds or the enzymes and so on. So we can have a clear idea of what will be the enzyme sequences that potentially could catalyze this reaction. So that is like a first step that help us to determine the ways of producing the target product. However, one problem that we observe is that this metabolic scope can become very large because we are working on the extended metabolic space. That is, it's not the basic metabolic network, but we are also considering, as I mentioned before, other new to nature compounds that are predicted by this generative system based on reaction rules. So that means that the extended metabolic space is huge, and often you find that the scope is contains hundreds or even thousands of possible pathways. So there's no way of manually enumerating those pathways, and this is why we develop, as part of the RetroPath suite, also another tool that will perform the enumeration of the pathways. There are different ways of doing this, but here in our approach, what we applied is elementary flux mode analysis, which basically takes the network that is as a hypergraph and then looks at the basic motifs that are contained in the network in order to calculate the elementary modes. In this example here, for instance, there are two basic elementary modes present, but then when we unfold these modes, what we found is that what there are four different or alternative pathways. So this is the type of graphical analysis that can be performed. And then, as I said, this can lead to hundreds or even thousands of pathways. So we need some way of ranking the pathways in order to be able to have some estimate of which pathways are going to be more preferential for us to implement. For that, we define here this function is like an objective function that contains multiple criteria that we combine together. Like, for instance, the reaction efficiency, the performance of the enzyme, or also the toxicity of the sequence, the toxicity of the intermediate metabolites. We have a tool for predicting this toxicity, or could also be ranked based on stoichiometric calculations. So as you see, it's more like a multi-criteria objective, and we need to be able to weight in the right or in the most optimal way each contribution of each term. And then there's another important aspect that came into this ranking function that I showed in the previous slide, that is the efficiency of the enzyme. This is one of the main bottlenecks that you will usually find in this type of pathways, because we have like multiple reactions that many of them are predicted, and we need to find a sequence of an enzyme that once it is expressed in the host organism is can be as efficient as needed for our specific pathway. So there is this tool that is called Selenzyme that basically what it does is to suggest enzymes or enzyme sequences based on multiple criteria, like you looking at the sequence descriptors, structural descriptors of the enzyme. So basically, you provide the reaction, and it will go through this large database that we have of several hundreds or thousands of reactions and also hundreds of thousands of sequences, and try to match the best candidates based on this criteria. We also have included AlphaFold or taxonomic criteria. This tool, it is implemented in different ways. You can use it as a command line, but very often what you will find is that end users like to use it through a web interface. So we have this web interface here that is very basic. You just need to provide the target reaction and specify the host organism and some other details that may be of interest for you. And with that, once the reaction is validated, the system provides this ranked table here of different enzyme sequences that are considered to be the best match for those reactions. So these tables are generated for each step in the pathway. And in that way, you have like a collection of candidates that can be tried, for instance, for directed evolution if they don't work initially as expected. So that's a tool that we decided to make it independent from RetroPath, even if it is connected, but because it happens often that this type of queries are performed afterwards, not during the pathway search, but afterwards when you want to increase the efficiency of the enzyme. But then I would like now to go into the second part of this presentation about the integration of the tools through the Galaxy SynBioCAD platform. So this platform has been chosen, the Galaxy system, probably you're familiar with it, is a way of generating workflows or pipelines that basically are interconnecting multiple tools. So in bioinformatics, you see many cases where Galaxy is used, and we found that also could be useful for us in metabolic engineering, even if that was something that at the time when this project was developed was not so common. But there was a collaboration between different institutions. You see here, like INRE, the University of Manchester, Liverpool, Imperial College, the University of Edinburgh, that together we combine our different tools in order to have a one-stop place where you can find all of them. And the workflow can be started, launched, and then just let it run for as much time as needed. So here you see an example of how this is performed. For instance, you start, this is the typical Galaxy interface where you select, in this case, the starting host organism that you want to work with, and then the desired workflow, in this case, retrosynthesis, which is basically RetroPath plus other things, but basically it's RetroPath. So if you look inside the workflow, you will see this typical interconnection of multiple steps. Well, it's a bit cut, but this is the first step is the RetroPath, but then it goes into the pathway enumeration, into the conversion into SBML, because one thing that we have tried in this workflow is to always generate data as outputs that are in a standard format, like in this case, SBML, that is the most standard format for metabolic models in systems biology. And then now, well, let's see this example of running the workflow for the host is Saccharomyces was in this example, and then we will use muconic acid, this building block, as our target compound. So after running the workflow, you see these different steps here that were successfully completed, and at the end, what it generates is just a collection of reactions similar to what we were discussing in my previous slides of this metabolic scope. So you see here, there's a very basic tool, but in Galaxy for visualizing the pathways like here, but also you can export this into other programs like Cytoscape and so on that allows you to do perhaps more friendly visualization. But this is good enough for us just to look at what are the pathways that have been found here. You see that there are plenty of them, around 25 or something like that. And for each one, you get the information about energies, fluxes, and so on. One other aspect that was integrated into this Galaxy workflow is the calculation of the thermodynamics in order to see what is the favorability from the point of view of the Gibbs free energy for that reaction. That's especially relevant in cases where you are working with reactions that are predicted. So maybe from an energetic point of view, this reaction is not going to be very efficient, or is even not going to happen at all. So that's something that we want to figure out, and for that, energy calculations were also introduced as a way of ranking the pathways. And as I said before, we have introduced different ways of computing fluxes for the pathway in order to see what is the maximum theoretical yield for the production. In my example that I showed you before, it will be of muconic acid in yeast. And here you see another example of that's one that we published a few years ago of producing pinosembrin, this flavonoid in E. coli, and there were 11 different pathways. And what also will give you the Galaxy is the ranking of the pathways, as I mentioned before. So just from the wet lab point of view, once you get this output from the Galaxy platform, it generates a collection of instructions with an experimental design like the one that you see here, telling which DNA parts needs to be combined and in which way, like which promoters, which plasmids, which RBS, ribosome binding site, and gene sequences in which order, and promoter exchange. All of these combinations are performed by the DOE or Design of Experiments module. So that this is an actual example of a case where we build this 16 library in a robotic liquid handling platform. And in that way, it was tested. No, you see here at the end, these are the four stages of the design, build, test, and learn. So in the test, it was quantified, and then in the learn stage, basically we observe at the different modifications or combinations to see which factors had the highest impact. And this is done in order to generate a second round of design rules to select those that most likely are going to provide you with the highest titers or levels of production in this case, it was of pinosembrin, of your target compound. So this is the basic strategy of the DBTL cycle. And what the Galaxies in BioCAD is allowing here is to have all of these different steps in one single place, so that you can go through them and then more easily automate the full pipeline. But it's true, that with this, I'm going to start now with the last part of my presentation. It's true that the DBTL cycle allows also for other types of applications or tasks that can be also performed, maybe not in a systematic way, but depending on the case, you could, for instance, do directed evolution of some of the enzymes. You could do some cell-free, or another strategy that lately is becoming quite popular or common in the biofoundries is to add some biosensors that will sense and detect the concentrations of the target compounds or intermediates in order to, for instance, apply it for screening or for dynamic regulation of the pathway. So in order to provide a solution for this biosensor design, we have another tool that has been published more recently that is called DetSpace. And it comes from a database that we have of different biosensors. Actually, what we have are transcription factors that can be allosterically activated by molecules, by effectors that are metabolites. And these transcription factors then can be used in order to build a full biosensor that is responsive and can, for instance, express a reporter like a GFP in the presence of the desired metabolite. The database is called SynBio, that also is open access. It can be downloaded if you are interested. It contains several thousands of transcription factors, and also it has some basic tools for comparison and similarity predictions. But then what we have done more lately is what we call DetSpace, which is another tool that in this case it performs systematic analysis for any compound of interest in the industry, and we are talking about several hundreds of compounds that are known to be the most relevant for the bioeconomy and for the circular economy. And we want to see if they can be connected with biosensors. And how we do this connection through metabolic pathways. This is one example here. There is again a web interface, but also there is an API, or you can call it through command line. And here, for instance, we have that our target is eriodictyol, and through a couple of steps, it can be converted into quercetin. That is a compound that can be detected by transcription factors, because we know this from our database. So what provides this tool, therefore, are designs of biosensors by looking at ways of interconnecting your target compound with compounds that are detectable. And all of these apps are available. Usually they have their tool, like a web interface, like in the case of Selenzyme, but also you can download them for your local installation using GitHub. In the case of RetroPath, because it is a 9 workflow, it's available at myexperiment.org. In the case of Galaxies in BioCAD, since it is a Galaxy project, you can either go to one dedicated repository, web server, sorry, that we have, that is this one, Galaxies in BioCAD, or you can install your own local instance of the Galaxy project by looking at the project website or the main repository for Galaxy. And for DetSpace, again, we have a web interface, or also it can be downloaded from GitHub. So basically, any of these tools can be either run in-house or through our web interfaces. So just to conclude, here I show you a DBTL pipeline for metabolic engineering to address the biomanufacturing challenges. And as you have seen, there are multiple tools and data sources that can be integrated through our scientific workflow platforms. These allow us to go beyond what is usually done in a wet lab, in an academic center, and more in the context of a biofoundry to perform an exploration of the full extended metabolic space in order to look for alternative and new to nature pathways and compounds. And then at the end, also, we have seen a tool that allows you to increase efficiency of the biomanufacturing process through biosensors for screening and dynamic regulation. And that's all that I wanted to show you today. Thank you for your attention, and I'm happy now to take any question.